Welcome to the lecture series on nucleic acid metabolism. So the question we're going to answer is, what are nucleic acids? Nucleic acids are biochemical molecules that are found within the nucleus of the cell. Now, the two major nucleic acids we're going to be looking at during the course of this lecture are the DNA and the RNA. In other words, deoxyribonucleic acid and ribonucleic acids. So what is the difference between these two? It's actually resident in the carbon sugar atoms. Now, the DNA and the RNA are polymers of nucleotides. So I've introduced another word, nucleotides. So what are nucleotides? Nucleotides are components of nucleic acid, and they form the building structure or the primary blocks for DNA and RNA. So let's look at the chemistry of nucleotides. Nucleotides are made up of nitrogenous bases, you have a nitrogenous base, you have a pentose sugar, and you have the phosphate group. So we'll go over it again. We have the nucleic acids, and then the major nucleic acids we're going to be looking at are the DNA and the RNA. Now we have the nucleotides, which are the building blocks for the DNA and the RNA. So what are the components of the nucleotides? You have the nitrogenous base, you have a pentose sugar, and you have the phosphate group. So now we're going to look at the components of the nitrogenous base. The nitrogenous base have the purine and the pyrimidine. So these are the two major structures we find in the nitrogenous base. Now the purine rings are more complex than the pyrimidine rings. So let's take a look at the purine ring. The purine ring is made up of a 9-membered structure. Beginning from this right side, this left, which is point 1. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's where we say it is made up of a 9-membered ring structure. So we are going to be looking at the sources of these carbon and nitrogen atoms in the purine ring, subsequent videos. Now let's look at the pyrimidine ring. The pyrimidine ring is made up of a 6-membered ring structure. So you beginning from here, the numbering, notice the numbering begins from here, all right, which is what we would say point 1, 2, 3, so this is 4, 5, and 6. So what are the examples of the nitrogenous bases you have in the purine ring? You have adenine and guanine. The adenine, which forms the adenosine triphosphate, which is the major energy currency of the cell. So we can say one of the major functions of the nucleotide is that it serves as the major energy currency of the cell, apart from the storage, all right, and the transfer of genetic information, which is the major function of the DNA and RNA. Now, for the pyrimidine rings, the examples you are cytosine, uracil, and thymine. All right. So these are the 3 pyrimidine bases that have the pyrimidine structure. So the cytosine, uracil, and thymine. When you have the pentose sugar and the phosphate group attached to this nucleotide base, all right, it forms the cytosine either cytosine triphosphate, uracil, all right, or adenosine triphosphate or adenosine diphosphate, depending on the number of phosphate groups. So now, what is the structural difference between the DNA and the RNA? It is, all right, majorly resident in the structure of the carbon sugar at position 2. The DNA have the hydroxyl group, all right, taken off, and you have hydrogen at point 2. Now notice, for the sugar, the numbering starts from here of the carbon atom. So this is point 1, 2, 3, 4, and 5. So for the DNA, this is hydrogen. Now for the RNA, you have this carbon 2 hydroxylated to give you the OH. So that is it for the introductory section of this class. We've understood the components of the purine and the pyrimidine ring, and we've seen the components of the nucleotide. Now note, when you have the nitrogenous base, all right, in combination with the pentose sugar, you have a nucleoside. When you have the pentose sugar phosphorylated to have the phosphate group, all right, at this point, carbon 5, all right, so you have CH2 here, and then you have this, and then you have the phosphate group, all right. So this is phosphorylated to give you what? A nucleotide. Drop your questions in the comment section below, and see you in the next class where we'll be looking at the metabolism of the nucleotide bases. Bye-bye.